Cool. Okay. Yeah. Welcome everyone to the first session of day 3 MPHC. I cannot believe it, like how time has been passing so quickly. Today's sessions are brought to you by Med School Coach, which provides pre-med and medical school admissions consulting services, MCAT and USMLE, complex tutoring, and unique products and experiences that help students become physicians. And today, we have our speaker, Dr. Henry Daniell, who's the Vice Chair and Professor in Penn Dental Medicine. Throughout his career, Dr. Daniell's passion has been always to improve global health equity and access. Inspired by issues of health inequity and global human rights, Dr. Daniell has developed a novel approach to orally delivering affordable biopharmaceuticals, eliminating expensive injections and the need for refrigeration. Let us welcome Dr. Daniell to speak on us on rectifying health inequity in pharmaceuticals. Welcome, Dr. Daniell.
Thank you, Michael. Thank you for the invitation. Let me see whether are you able to see the screen, Michael?
I am not. Pretty much like it should be the same share screen process though.
Okay. Are you able to see the screen?
Yep, there we go. Okay.
Okay, great. Good morning, everyone. It's wonderful to share and painful to point out the challenges in global healthcare inequity, inadequate access. So today I'm going to remind you, most of you are aware of the cost of healthcare, but also as you start your careers, these present great opportunities for you and your generation to change the status quo. Let me start with the Okay, so let me give you just a few examples of the cost of healthcare. For example, last month, FDA approved gene therapy for hemophilia. Per patient, it is $2.9 million. This was published in Wall Street Journal, June 29th. A few months before that, FDA approved gene therapy for hemophilia B, which is another form of hemophilia, which is $3.5 million per patient. If you look on the right, you will see that what is injected is 8 milliliters of virus. So essentially, to prepare 8 milliliters of the virus, it's not really the reasonable to charge $2.9 to $3.5 million. This is, you may think that this is a rare disease, but this happens in all common disease. Most common is the diabetic epidemic, which is a silent epidemic because most of us were inundated with COVID. Number of deaths in the during the entire pandemic is 1.1 million deaths in the US. But guess what? Diabetes kills 6.7 million deaths every year. And insulin was invented 70 years ago, and insulin as injection was introduced 60 years ago. And if you look at the global prevalence of diabetes, it's all over in all the countries. The numbers change. Some countries have more diabetics than others. If you look at the right top panel, you would see the healthcare disparity, inequity. If those with low income or lower middle income, this is actually 10-year-old data. The data now would be a lot worse. They're out of luck compared to those who have high income. This is 3, 4, 5 times greater chance of the death related to diabetes. In the US, the Medicare beneficiaries, who are usually those who are eligible for Medicare, are poor based on the economic status. Out-of-pocket expense was over $1 billion a couple of years ago, but it was $235 million in 2007. So as you can see, the cost not only of insulin, other top 10 protein drugs, is $200 billion, which is 3/4 of the global GDP. And 1/3 of the global population earn $2 a day, but the insulin pump costs $6,000 to $12,000. So you can see the disconnect between what is needed and what is available. So I'm going to point out technical reasons for this. One of that is that injections are invasive. Some other protein drugs are implanted surgically, even injected into the eye. One of the things that we have advanced is avoid injections by delivering the protein that are made in plant cells. Why plant cells? Because we eat them all the time. So we don't have to show that any of the plant protein is immunogenic because we eat them, body has developed tolerance to it. So we produce insulin and all the other proteins in plant cells, and we freeze dry them and follow the FDA guidelines. And it could be delivered in capsules, or for many of the oral viruses and oral pathogens, they can be delivered using a chewing gum. If you want to learn more, there is an article that's going to be published next week in Nature Biotechnology, which is a top journal in this field, where we show how greater than 90% of drugs approved by FDA in the past 7 years are injectables. There are few examples of oral, sublingual, or ophthalmic. Most of them are invasive. But if you make them in plant cells, then that is friendly as far as delivery, non-invasive, but also it reduces the cost. So let's look at the cost. Insulin, if you take insulin, human insulin gene is cloned, and it is produced in bacterial cells, E. coli cells, or in yeast cells. And once they are grown, they have to be grown under exceptionally sterile conditions. Air needs to be exchanged according to FDA guidelines every 30 seconds. And if you look at these machines, they cost about $500 to $900 million. And then humans are allergic to these proteins, so they need to be purified. Each of these columns is disposed every single day, and this is $10 million column. And therefore, after purification, before injection, insulin pens cannot be stored for long because the proteins are unstable. So all these things add to the cost, and the regulatory stacks needed for approval cost on an average about $2.5 billion to introduce a new drug. Whereas the new technology that using plant cells, the cost of developing a drug is somewhere between $10 to $25 million. Because it's orally delivered, the cost is less than 5%. In this case, the peanut allergy drug developed recently, commercially available, is about 3%. When I came to this country almost 40 years ago, one of the first things I did was call my mom home to say that I reached here. There was no direct dialing from US to India, and that call cost me $75 because it went through the operator. That was almost 10% of my salary. But today, with WhatsApp, Zoom, or Skype, it's all free. I can see them. So that kind of a technology has not, revolutionary technology has not been advanced in medicine. We have gene therapy, but not towards addressing the cost. Insulin was injected 50 years ago, it's injected till today, and 95% of the drugs approved by FDA are injected. The worst point is that injections are not suitable for drug delivery, especially in diabetes, because when you inject insulin, it gets diluted in blood, and then it rapidly decreases blood sugar levels, leading to hypoglycemia. You will see that in a few minutes. Whereas natural insulin is released from the pancreatic beta cells, it's released around the liver, which when goes to hypoglycemia, liver glycogen metabolism takes place right there. So it's injected at the wrong place. Whereas when it's natural insulin is released from pancreas, it doesn't go to hypoglycemia. So in a sense, then one should pay attention to the delivery method, and the drug is the same, but the delivery should be different in order to address the cost. I don't want to go into nitty-gritty details on this, but I just, in a few seconds, I want to explain how this is done. We clone the human genes, we shoot them using a gene gun, and we monitor the cells that capture the gene. We take them to get the seeds, and the seeds will have the genes integrated into the plant genome forever and ever. And we give the seeds to commercial facilities. Once leaves are harvested, they are freeze-dried, powdered, and made into capsules or chewing gum. And there are commercial facilities. There's a lot of them, indoor facilities, where we give the seeds and we will get the biomass product. The slow process is getting FDA approval. If you can see here, this product, the angiotensin converting enzyme 2, was developed 10 years ago. We received the FDA approval last summer. And so we have to go through the rigorous regulatory process to show that it's free of bioburden, moisture content, drug stability. All of these things need to be done to meet FDA requirements. Once we do this, then it's wonderful to receive an email from FDA that we have approved your drug, you can give this to patients. So this is what happened. What's the breakthrough here is that this is the first engineered human protein in a plant cell, first clinical product that's approved without need for purification or need for cold chain, because plant cells are stable for many years and protect the protein. And the scale up, 1 million A's to gum can be made from 1 kilogram of the plant leaf powder. And this enables both oral and the topical delivery. Again, I don't want to go into the details. Once we get FDA approval, we have to meet all those FDA requirements for the description, microbial burden, drug dosage, efficacy, potency, and so on. So that's the background of this technology. To give you some, I will illustrate the power of this technology with just few examples. I will start with COVID. Many of you, I'm sure, know someone who had COVID. And how does the virus affect humans is that it binds to this enzyme ACE2. And as you can see in COVID patients, which are shown in red dots, the ACE2 enzyme activity is completely knocked out compared to the healthy volunteers we examined. We examined under all kinds of conditions. And once we did that, I came up with the idea that if virus binds to the enzyme, can we use the enzyme to trap the virus so that we can decrease the infection? And that's what is shown in the top portion. So we expressed ACE2 in lettuce. We have got FDA approval, and it binds to the virus and it traps the virus inside the gum. Or we had another viral trap protein, it's called lectin, which makes the virus aggregate among themselves, and therefore it cannot enter the human cells, or if it escapes and enters, it is prevented further entry into the nucleus at the endosome level. So essentially, the concept is very simple. You produce a viral trap protein, it acts like a flypaper, and it essentially traps the virus inside the gum. How does this work? We have another Penn faculty developed a method to see COVID virus, SARS-CoV-2, in the saliva, in the swab samples. These are called microbubbles. Each virus is a bubble. So once you add the gum powder to it, the bubbles disappear. And essentially, as I showed in the previous slide, it traps a virus, and whatever is remaining, if it enters human cells, they will be blocked further at the endosome. And this is what the clinical trial right now, the recruitment and other things in progress. We got FDA approval by taking patient sample, adding gum, and showing that it works. Now, with FDA approval, we can give it to patients. We are doing it backwards. We are taking saliva and swab samples before giving the gum. After they chew the gum, we take the sample again. And that's the trial that's in progress. But is this only a pandemic product? No. We can do coronavirus. We can do flu virus. There are potent flu strains. We can do oral herpes. We can do a number of different viruses. In the next slide, I show how several different viruses can be neutralized using this gum technology. The amount of the gum needed is very small. We get several hundredfold more, and the gum slowly releases, and there is a steady release of this protein during chewing up to 30 minutes. But typically, the clinical trial is done for 15 minutes. Can this do neutralization with other pathogens? Because yesterday I was reading that in coronavirus patient death, people who had coronavirus also had the yeast, Candida albicans, and their death rate is 4 times higher than those who didn't have the yeast infection. So we can use enzymes to kill the yeast and the bacteria that causes the dental plaque. So here, the red color is the plaque, and the green is the bacteria hiding under the plaque, and the Candida albicans is the one which is long filaments. Once they chew the gum, you can see that the plaque is dissolved and the bacteria and yeast are killed. You can see the yeast spore cannot germinate because it is inactivated. So these are examples of how topical delivery using chewing gum can address a number of diseases in the oral cavity, whether it is virus, bacteria, or yeast, or fungus, can be done. But more importantly, we need to deliver drugs like insulin so that it could be taken orally. The principle behind the oral delivery is that when you express the insulin in a plant cell, human enzymes cannot digest the plant cell wall. So the insulin is protected from acids and enzymes in the stomach. But then when the plant cells reach the gut bacteria release enzyme to break down the plant cell wall because they get amino acids out of them, and the drug is released, then it is absorbed. If you see on the right, this concept is demonstrated using a green fluorescent protein inside a plant cell. You can see on the right intact plant cells inside the gut, and then you see it on the gut epithelium because the bacteria broke it, and then the GFP has entered the cell. So here, then we compare the current insulin delivery methods, whether it is a pump or pen or injection, to the oral delivery. And when we do that comparison, you can see that the red is any form of insulin injection. It rapidly removes glucose, that the body goes to hypoglycemia, and then the liver has to pump glycogen to bring it to normal levels of sugar. Whereas natural insulin, which is in yellow color, doesn't do that. It is slowly released, and it is released around the liver, and it doesn't go to hypoglycemia, but it gradually removes. Oral insulin, which is in green, made in plant cells, follows the same kinetics as the natural insulin. Even more important is that after 60 years, natural insulin is not what is delivered in the clinic. The injectable insulin is missing what's shown in red here, which is the C-peptide. Insulin has 3 components, A, B, and C, but 60 years ago, they didn't know how to make 3 peptides, and it is still sold as incomplete insulin for 60 years. So our insulin and natural insulin have all 3 components. Finally, I'll show one more example how the oral delivery can treat COVID or hypertension patients. I showed you the slide before how ACE2 enzyme is key for human health. When this enzyme is less, then you can see the right ventricle enlarges, it causes hypertension, and then it cannot pump. When we give this orally for 2 weeks, pumping is restored greater than 95%. So in the end, the restoration is dramatic. That's because ACE2 enzyme is protected in the stomach and delivered to the gut. So in summary, then the technology is that first problem is the cost of drug development. For example, the gene therapy, I told you what the $3 million cost, because the development cost is so complex for injectables that it takes $2.5 billion. But then when you reduce the complexity of the drug delivery, it can be reduced to 10 to 25 million. Second, we can completely eliminate the fermentation process because plants can be grown in greenhouses. You don't need this $500, $900 million. Finally, last year, you may have read 20 million donated doses in Africa were destroyed because they didn't have cold chain. But for us, the plant cells are stable at ambient temperature, and therefore it can be stored. There is no need for cold chain. So in a sense, what's needed for, especially I present this challenge to the next generation, that it's not only developing new drugs, but one should pay attention to their affordability and global access. And this is going to happen only through technological revolution. Moving money around could help, but ultimately, just like the cell phone technology that I mentioned earlier in the talk, this is what is needed to change the status quo. For those of you looking for career guidance based on my years of experience, that the one guidance that I can offer you is please follow your passion. When whatever that I have done in my career to reduce the cost of drugs and technology development, I thought about it when I was at your stage in my career. So in my 20s, I dreamed about developing drugs that are affordable, because when I grew up in India, I used to see small coffins in churches. I used to ask my dad, why is this happening? And he used to explain that they cannot afford the drug, that drugs are available in Western countries. So when I came to this country, I made a commitment that if I get a chance to develop, I will focus on the drug delivery. Today, I have great news to share with you that the product has been developed, several products have been developed, and the product has been approved by FDA. And it's good that there are actually the opportunities that were provided to me. I was able to utilize this. And it's also nice to see recognition of the technology and with the fellows of National Academy and all those things and awards. But please don't get carried away by these kind of recognitions, because ultimately what needs to happen is the technology needs to be developed and taken to people. I've been fortunate to be among the top 3 eminent scientists at Penn with senators, gave a TED talk on April 1st. You can look them up. And it was very challenging to give a talk. The second from left is Anthony Russo, who I'm sure you're familiar with Modern Marvels. He gave the talk, and then I had to follow him. He showed the 3D movies of the trailer. And but ultimately, the greatest thing for me is developing careers of hundreds and hundreds of scientists who worked in the lab, and their hard work and commitment actually is the result that you see here. And this is for 4 decades. So many careers have been spawned, and many of them are professors in medical schools, are directors at companies, pharmaceutical companies. And all of this is possible because it requires lots and lots of funding from federal agencies and pharmaceutical companies as well. With this, I close, and I want to thank all those who contributed to this study. And this is the latest lab group that I have, whose results that I presented here. At this stage, I will stop and answer your questions. Thank you.
Great. Thank you so much, Dr. Daniell. That was certainly inspirational for me as personally who has been always just wondering so much about like how do we rectify health inequity and like how much like the system like how much of the status quo that we need to change nowadays. It really inspires like people to ponder about like how do they leverage their unique maybe key opinion leader positions if they're pursuing a career in healthcare to actually like just make sure that people are aware of those issues and then drive the society toward altogether a better direction. But yes, thank you so much for such an amazing presentation. And yeah, I guess I'll just like dive into attendees' question real quick. The first question is actually that what would you say is your proudest accomplishment from your perspective throughout your like entire career in translational sciences?
I still, although FDA approval of the drug would be the proudest accomplishment, I still consider that it falls short of accomplishment because it still needs to go to the people. So I would consider that once it reaches the market, once it reaches poor countries, developing countries, and once it's made available, then I would consider that as an accomplishment. I think we are almost at the light is at the end of the tunnel, and it's been a 40-year tunnel. It has taken a long time to get here, but the good news is that we are not talking about just insulin or one product. We are talking about all major diseases, inherited metabolic diseases, and also infectious diseases. So there is a lot more work to do. And I want to take this opportunity to inspire the next generation, you guys, to take the lead now and change the status quo, because it is unacceptable for one patient to pay $2-3 million for a one-time treatment. And so that's where the accomplishment is needed. But so far, our greatest accomplishment would be getting the FDA approval for the product for giving to patients.
Yeah, I'm wondering, like, when it comes to your career, basically to pursuing like not only on from like rectifying health equity lens, like what inspired you to pursue actually a career in translational science, and how, I guess, like targeted toward our pre-health audience today in the room. How do you think that really the field of healthcare and academia should go together, Ed?
Thank you. So as I mentioned, my inspiration came from growing up in a developing country, seeing people die because they could not afford the medicines, whether it is vaccines or whether it is insulin. In fact, my dad died of diabetes, and insulin was not available in India at that time. So I had a lot, and I lost my dad when I was 11 years old. So these are painful experiences to see and ask the question, why is it that it's not available? Why is it that it's not affordable? So that's where my conviction started when I was young, when I studied in college. And that as far as those of you guys starting at the beginning of your career, this challenging yourself, finding passion to do something that you are passionate about. And it could be any disease, it could be anything that kind of inspiration is a self-motivator. It helps you to drive. You're not waiting for somebody to pat on your shoulder and say, hey, you're great, as opposed to that, well, this is my goal. I really want to move forward. And in that process, you will be impacting the careers of a lot of your colleagues and your students. And so this takes an entire village. I'm not only talking about doctors or scientists. It takes an entire village to take a product from the lab to the clinic. It takes healthcare journalists. It takes people to communicate the innovation to investors. And it takes public to get to know this. And scientists are not trained to do that. And it takes nurses, it takes healthcare professionals to pay attention and bring this to the people and to the attention of those who make these policies. So it takes an entire village to move, change the status quo.
Yeah, I honestly agree. Like, it's only through all stakeholders in the society that work together that we were able to change the status quo. Yeah, actually, Samantha here is asking, like, are there any ways to get involved in your work or maybe your mission?
Absolutely. You could get involved even remotely, because a lot of these days, a lot of research are done with the tools for artificial intelligence, information gathering, and helping promote new technologies compared to current technologies with the local community, and educating the local community, and talking with the local hospitals, and directly participating and moving things forward. Obviously, this is not a one-man show. This takes an entire community, but now essentially the ball is in your court. You guys not accept the status quo that it's okay to have $1 billion copay for insulin of the poorest people, the Medicare people. It's just not acceptable to have $6,000 to $12,000 pump when 1/3 of the world earned $2 a day. So things need to change. Just like technology revolution changed the communications, cell phone, and all that, biomedical sciences need affordable technology.
Right. Okay. Cool. I guess like this person is actually asking a similar question. So like, how do you think like particularly professions in the health industry can contribute to like lowering health disparities and advocating for university and affordable healthcare system?
Absolutely. It can be done at every level. It can be done in the clinics. It can be done in the hospitals. It can be done by nurses. Then, first of all, you guys can share this information with your local clinical community and healthcare professionals and educate them that, because all these requires policy changes. And that means it's everyone getting involved and connecting with people whom you know, and then point out the disparity and how long this is going on, and that the just like everything else, climate change and everything else, public education and public understanding, and you guys are the ones who are very well versed with social media. And it's not my generation, but it's your generation. So you are talented in communications, and you could learn more about this. Again, I'm not promoting just plant technology. Any technology that can make drugs affordable. So I'm not saying that this is the only technology. This is one example of a technology where cost can be addressed.
Great. Thank you so much for that answer. There's another attendee that we're wondering like what can we do now, like particularly as undergrad or pre-health students, or just like at a more junior position in our careers right now, to get involved and decrease global health inequities. Like, are you aware of like any opportunities with any particular like organizations or stuff like that?
Yeah, I have a lot of, in fact, if you look at the names that I listed here, quite a lot of them are undergraduates. And so they are authors. Some of them are in their 4th year of undergraduate education. So there are websites, definitely at Penn. There's websites for the faculty research is posted at the undergraduate research website. And so most universities will have they describe briefly, but then you guys go to Google Scholar, type in my name, you will see all the publications and so on. So there are lots of research is really encouraged at the undergraduate level, because those are all the formative years, and clinicians eventually, it's great that they save lives, but at the same time, after anyone starts a clinical career, that's another 40, 50 years of practice, right? So at that time, if one keeps up with the changes in technology, innovation, this helps them to keep up with the changing technologies as they get into practice.
Gotcha. Thank you so much. And let's pivoting back to the your work a little bit more. There's a person that was asking like, how far do you think all those new developments like are basically before they can be provided in poor countries where basically there are little health related improvements?
Right now, this is under evaluation in clinical trials, and once FDA approves and release the product to the public, that can happen after phase 3. Then it's a question of integrating with the developing country government bodies. Like, for example, India has a for poor students, they have a midday meal program. So this could be integrated into such government programs where these chewing gums can be given to remove their plaque, to save their tooth infection, tooth loss, and all those things are all the viruses. The oral herpes, there is no vaccine. It's transmitted through kissing and coffee cups. So this, once it is developed, the government agencies, World Health Organization, all these things need to be involved to facilitate this process. As you can see, the system didn't work well because developing countries and poor middle income, low income countries only got 4% of the vaccines. The developed countries got the rest. So there are many levels where the young generation can get involved and make this health equity technology. It's a starting point, but the end point of reaching those who need this the most will happen with the entire village.
Gotcha. Thank you so much for the answer. And actually, Angelina here is asking a technical question. So she heard that plant cells are more tolerant than animal cells to genetic mutations, such as aneuploidy. How susceptible are plant cells carrying the gene for the drug to spontaneous mutations that would render the drug inactive or even harmful? And yeah, I guess that's what she's curious about.
Sure. So what we are delivering are plant cells that are freeze-dried. So they are not living cells. So they cannot be mutated because there is no water, there is nothing in the plant cell. And not only that, we insert the gene into the chloroplast genome, which is highly conserved. After billions of years, the chloroplast genome still hasn't changed. Whereas the nuclear genome changes because there is Mendelian crossover. Whereas chloroplast genome is maternally inherited, it's like mitochondrial genome. And therefore, the mutations are almost close to zero. And not only that, the cells are not living, and therefore they're not exposed to any mutation.
Gotcha. Okay, with that being answered, that concludes our session today. Thank you so much, Dr. Daniell, for such an inspirational presentation on your work on how to rectify basically health inequity as a scientist. And I'm sure we have a lot of takeaways, all of our audience in this room, on like, how do we pursue that mission in the future of in our careers as well. But yeah, again, thank you so much. Let's send a biggest virtual applause to Dr. Daniell. And yeah, thank you so much for speaking at our conference as well.
Thank you so much, Michael. Thanks for the opportunity. Appreciate it.
Yeah, definitely. Thank you so much. Our pleasure to have you. For our participants in the room, we are going to transition to next session, for which Dr. Moore is actually going to be sharing on how climate change, as well as like public health, could be a sort of a traditionally overlooked area in healthcare, and whether how it's very important and plays a role in all of our careers as future healthcare practitioner. So yeah, stay tuned to that. And I'll see everyone in a couple of minutes.